hemoglobinuria in sheep and goat, the causes and diagnosis. Physiological agents of hemoglobinuria. So first, it can be divided into two: infectious or non-infectious causes. So bacillary hemoglobinuria, Clostridium perfringens, then erythrocytosis, then babesiosis, and leptospirosis. These are the important infectious causes. Other infectious causes are also there, but these 5 important diseases which cause hemoglobinuria in case of sheep and goat. Bacillary hemoglobinuria. It can be defined as it is an acute fatal disease of sheep and cattle. So goat are also affected, but sheep is more afflicted with this. And it is characterized by hemoglobinemia and hemoglobinuria. As it is indicated by this name, that is bacillary hemoglobinuria. So hemoglobinuria has to be there. So this is a disease of sheep and goat and cattle. There are many factors which cause hemoglobinuria. One important diagnostic factor in this case of hemoglobinuria, bacillary hemoglobinuria, is the season. Actually, it is more common in summer and autumn, and this is particularly seen in at the time when the fluke population is more in the animals. So whenever there is endoparasitic infection and whenever there is excess of starvation, when the animal's immune system goes down, so that time you will find this hemoglobinuria, and you can 80% diagnose it as because of Clostridium hemolyticum, because of the season and because of the prevalence of flukes in that particular time period. So what happens, the flukes, they are eggs of the flukes, they are present. The animal, they go, they take the snails, and this metasaccharide enters into the body and it migrates to the liver. Now this fluke, it develops in the liver, in the bile canaliculi, and in the liver hepatocyte, in the hepatic tissue. So this, while traveling, it creates a path which causes damage to the cell or to the tissue. So this Clostridium hemolyticum, it is there in the body, and along with the fluke, along with the track of the fluke, it goes and it settles. And since it is getting a nutritive media of the damaged cells, so it will proliferate on that, and it will lead to damage to the liver and to the Kupffer cells, and this migration of immature liver flukes and other parasites also, that causes the actually that acts as a nidus of proliferation for this Clostridium hemolyticum bacteria, and there will be necrotic hepatitis, and there will be parenchymal damage, and the bacterial spores, they start germinating, and that lead to the hemoglobinuria. So this bacteria, as I said, it proliferates and it releases the exotoxins. Clostridium have that tendency to release toxins. So they release exotoxins, including the beta toxins, phospholipase C, and this causes hepatocellular necrosis, and this also leads to intravascular hemolysis because of shortage of certain types of proteins, amino acids in the blood, which are synthesized by the liver. Since the hepatic tissue is damaged, these amino acids or these proteins are not being formed there, and that leads to hemolysis, and this again hemolysis leads to anemia and hemoglobinuria, and this is the characteristic of this disease. So migratory tract of the fluke actually precipitates the disease. So while diagnosis, we can go for identification of the fecal sample. If you are getting hemoglobinuria, along with that, if you are getting fluke eggs, so 80 to 90% diagnosis is that this is because of, and if there is some pain in the abdomen, especially on the right side of the abdomen where the liver is situated, so you can palpate the animal, and because the animal will elicit pain, and if you can see a enlarged liver which can be easily palpated at the hypochondric region and ahead of the last rib. So the liver is enlarged, actually liver's anatomical position is hypochondric area, but in case of enlarged liver, you can see a much amount or much mass of the liver, it is beyond that margin. So one thing is pain in the abdomen, then you can find the eggs of the fluke in the fecal sample, and then hemoglobinuria and the season. Along with that, you can culture the organism. If you can take a biopsy of the liver, you can do that, and even if you can go for culture of the fecal sample, you can get the Clostridium organism. So this is about the diagnosis of bacillary hemoglobinuria. This is a liver which was afflicted with the liver fluke, and it had bacillary hemoglobinuria. So you can see multiple tracts of the liver and necrosis of the liver everywhere, and thickening of the bile canaliculi. See here, the periphery of the bile canaliculi, it is whitish in color. That means it is fibrous. Usually it is hollow structure, but when you see a peripheral ring of white color, that means there is calcification and fibrosis. So this is a typical liver of bacillary hemoglobinuria affected liver.